All right, hello everyone. In this video, we're going to be talking about membrane transport. So if we consider a lipid bilayer, right, and it's hydrophobic in nature, so if we think about ions or substances that are polar, the movement of such substances and ions across the membrane itself can act as a barrier for the movement of polar and charged substances. So what we discuss in this chapter is transport proteins, proteins that facilitate the transport and the movement of substances across the membrane. All right, so this is really governed by free energy change, or the chemical potential of a given substance and its movement across the membrane. It depends also on the concentration of that particular substance on either end of the membrane. And if we're considering ions, it also depends on the membrane potential. So we could think about the chemical potential, which depends on its chemical potential at a standard state, plus R, the ln of the concentration of A, right? And its movement across the membrane. So if we're considering the difference between the concentration in, or between the chemical potential in and out as it is moving, then we could rewrite it really as delta G of A, or the chemical potential, is equal to RT, the ln of the concentration of A inside the cell versus the concentration of A outside of the cell, right? And we call this the chemical potential. All right, now if we're considering ion, we also have to keep in mind the membrane potential and differences within the potential, and that depends on the charge of the ion, and that is given the symbol Za. It also depends on the charge of a mole of electron, which is given by Faraday's constant. Okay. In the membrane potential as well, right? And then instead of calling it a chemical potential, it is known then as the electrochemical potential. All right. So for substances that are charged or polar or ions, they cannot really diffuse alone across the membrane, and then their diffusion is facilitated or mediated by specific proteins, right? Okay, so there are a number of different types of membrane transport. One is known as a non-mediated transport, or what we refer to as a simple diffusion, right? Where molecules could simply diffuse across the membrane. One is known as a mediated transport, and those occur via the action of proteins. So you have a specific protein that would be embedded within the membrane. And there are two types, either passive mediated transport, which is going, within a concentration gradient, going from the low concentration, going from a high concentration to low concentration. So those do not require the input of energy. A different type of transport is known as active transport. And the process itself, or those substances, are moving against their concentration gradient. So they are moving from high concentration to low concentration. And that process is endergonic. It is not favorable. So active transport is coupled with an exergonic process, typically the hydrolysis of an ATP molecule. All right. So an example of a passive mediated transport. So if you think about a hydrated solute, all right? So you have a solute and it's hydrated, right? So here's our solute, and the solute is hydrated, right? And such solutes can pass through biological membranes by basically dehydrating, right? And that shedding the dehydration shell is energetically unfavorable. So if you notice in simple diffusion, the energy barrier, or delta G, is very high. However, in the presence of a transport protein, right, then once of a sudden that energy barrier is much smaller. And what is happening is that the solute, or that substance that is about to move across the membrane, right, it is shedding its hydration shell of water, which again is energetically not very favorable, right? But then as it passes through the membrane, that particular solute will form non-covalent interactions with the lining of that particular protein, which will basically make it energetically favorable and lower the overall delta G. And we call such, an example as facilitated diffusion, right, through the aid of a protein. So that's the difference between a simple diffusion or a non-mediated diffusion versus a facilitated diffusion. All right, more energetically favorable. All right. So in passive mediated transport that do not require the input of energy, we're going to be talking about a number of different examples. Their thermodynamic principles are the same, but those proteins really differ based on their structure, their mechanism, which ions they're selective for, right? So we're going to discuss some of them. Right. One example are known as ionophore. They're smaller molecules, right? And they allow ions to diffuse, basically, just like the name implies, ionophores. They allow ions to diffuse across the membrane, right? And based on their concentration on either end of the membrane. There are two different types. One known as channel-forming ionophores, where the ionophore, which again is a protein, is forming a channel across the entire membrane, right, where ions can diffuse through depending on the concentration on either end. Another type are known as carrier ionophores, and those basically also increase the permeability of the membrane for specific ions, but ionophores can bind a selected ions, or carrier ionophores bind a specific ion on one side of the membrane, and then that protein or peptide or that molecule can diffuse across the membrane and release the ion on the opposite end of the membrane, right? So those are known as carrier ionophores. They bind, they carry that ion, they diffuse through to the other side of the membrane, and then they release it, right? Channel forming have a channel where the ion is passing through. So one perfect example of an ionophore that is very selective towards potassium ion is known as valinomycin, right? Specifically binds potassium ion and transports it across the membrane at a very high rate, right? It transports 10 to the 4 potassium ions per second. So if we take a look at valinomycin, it is composed of both D and L amino acids, all right? We have L-valine, dihydroxyisovaleric acid, D-valine, and L-lactic acid, and it has two different types of bonds. All right, it has two different types of bonds. It has both peptide bonds and ester bonds present, right? Okay. And so those are the different types of bonds. And what it also has is those isopropyl group and the methyl group on the L-lactic acid. And those are basically pointed towards the outside, which makes it soluble within the hydrophobic lipid bilayer and thus can move through. And then we have potassium ion that fits snugly within the core of valinomycin, right? And it is a cyclic peptide where it has 3 different repeating units of that sequence, all right? And then they're finally connected to one another. And that's what this line is basically indicating. So those 4 repeat 3 different times, and then they're connected, so we have this cyclic molecule, right? So then it's soluble within the lipid bilayer. So it fits potassium ions snugly, and that potassium ion is coordinated to 6 different carbonyl oxygens of valine, right? So here's the carbonyl oxygen of valine, right? You have 1, 2, and you have 3 of those, so those are the 6 carbonyl oxygens of the valine residues. So it fits right in. Now, it can transport, so if you think about potassium versus sodium, they're both within the same group of the periodic table. Potassium has an atomic radius of about 1.3 angstroms, whereas that of sodium is about 0.93 angstroms. So that small difference in the atomic radius makes potassium fit so tightly or snugly within valinomycin, and it can transport potassium 10,000 folds better than it does for sodium. So it's very selective, right? Okay. Another example is porins, and we had talked about those on our previous chapter, which are beta barrel-like structures where you have a number of different anti-parallel beta sheets that are wrapped around, where at the end you have the N and the C terminals in close proximity, and you've kind of formed this aqueous channel right in the center, right? And the size of that channel and the aligning of amino acids within it dictate what type of substances can pass through. And one example is maltoporin, right? It's a bacterial outer membrane that facilitate the movement or the diffusion of maltodextrins. And maltodextrins are basically glucose molecules that are linked to one another via a 1,4 linkage, right? So they are formed from the degradation of starch. And if we take a look at maltoporin, it's a trimer, so it has 3 different subunits. Each of its subunit has 18 anti-parallel beta sheets, right, that are wrapped around to form this central channel. And then you have these loops within it, all right, that are basically forming that lining of that barrel, or the, not the lining, but the entrance of that barrel. You have those loops. Right. So it has a channel that is about 5 angstroms. It's very selective towards near planar groups, and that's why it can transport glucose molecules that are linked by an alpha 1,4 linkage, right? And the lining of the channel is hydrophobic or aromatic amino acid residues, tryptophan, tyrosine, tyrosine, tryptophan, tyrosine, and you have in phenylalanine. So you have those 6 hydrophobic residues that fold in a somewhat of a left-handed helix, right? That resembles that of an alpha amylase. Okay. And what happens is then the glucose molecules will stack on those aromatic side chains along what we call a greasy slide. This is known as the greasy slide. And then the hydroxyl group of glucose are involved in hydrogen bonding, right? And then there's also a specific tyrosine residue on the opposite end of that greasy slide, which permits only the passage of near planar groups. So it acts as a steric barrier, right? So it's very selective. And that's an example of maltoporin. So it would be present within the membrane and allows the transport of maltodextrin. All right, so some are ion channels, which basically open and close in response to a stimuli, right? They will close and open depending on the concentration in either end, right? It could have 2 gates that open and close, or 1 gate that opens and closes, right? So, and what it does, it allows the rapid movement of ions across the membrane to balance the osmotic pressure, signal transduction, or changes in the membrane and the chemical potential of the cell. So if we think about mammalian cells, the concentration of sodium and potassium on either end. If we're thinking about passive mediated transport, or proteins that are allowing the transport of ions across the membrane within their concentration gradient going from low to high, then it will transport, for instance, potassium from outside the cell, from inside the cell to outside the cell, from the higher concentration to the lower concentration. Later, when we talk about active transport, we'll talk about the other way around. But meanwhile, we're talking about passive mediated transport within a concentration gradient. So going from inside to outside. One example is the potassium channels, all right? And they specifically selectively transport potassium across the membrane, and they have about 10,000-fold higher permeability to sodium than potassium, and that does have to do with the atomic radius, just like when we talked about valinomycin a few minutes ago. All right, so one example is the KCS, a potassium channel. It's a homotrimer, tetramer, so it has 4 different subunits, and each subunit is colored in a different color, right? They're composed of 2 alpha, longer, almost parallel, nearly parallel alpha helices, and a shorter helix, right? So you could see you have 2 longer helices, and this is the shorter one. You have 2 longer helices, and here's the shorter one. If you take a look at the blue, we have a longer helice, another long one, and there's a short one right here. So it's composed of 4 different subunits, right? Those alpha helices are within the membrane, right? A shorter part of it is in the inside, and then you have a loop towards the outside. Okay. So we're going to talk about it, but it has this selectivity filter that only specifically would allow potassium ions to pass through. So if you notice, it has this four-fold symmetrical structure, and the ions will pass right in the middle. Okay, so if we take a look at it, it's about 45 angstrom long of a channel. It has 4 anionic residues towards the entrance of the channel, which will attract positively charged ion, which is the case of potassium. But that particular part is 6 angstroms because potassium comes in a hydrated form. Okay, and as it passes through, that center part is about 10 angstroms long, and it's lined with hydrophobic residues, and that's basically the yellow, right? And they come in a hydrated form. And then as they exit, that's where we have the selectivity filter, right? Which has a characteristic sequence TVGYG. It's a signature sequence in potassium channels. And then it is much narrower here because it basically shed off its hydrated shell, right? So if we take a look at what is happening, you basically have this potassium ion that comes in a hydrated water molecule, and you have this larger cavity, right? It goes through, and then as it sheds its water of hydration, what stabilizes it or causes it to move across that protein channel is that the potassium ions will basically be held together in lieu of the water with the backbone carbonyl oxygens. And what is shown here is 1, and then it moves to the next, and then it moves to the next, and what it's doing, it's basically coordinating to the carbonyl oxygens. And because of the size of potassium, it would basically transport potassium. Potassium fits snugly, sodium doesn't, right? So it has these alternating sites that are occupied, and it's the carbonyl oxygens that will act as a cage and precisely replace water hydrating shell and cause potassium to be transported out, right? Okay, so we will talk about this on our next video because of time.